moving on to the stomach. So we have taken in the food, we have chewed up the food, we've swallowed the food, and now we're going to break down the food into its liquid form in the stomach. So we're going to go over the anatomy of it, our different secretions in the stomach, how we actually create hydrochloric acid in the stomach, different enzymes, and then our 3 phases of gastric function. So let's look at our stomach anatomy. So our stomach is very muscular because we have to move the food around, mix it with our different gastric secretions in order to go ahead and break down the food from a solid to a liquid form. So it's going to mechanically break up the food, so that's the churning of the muscles, help to liquefy it, and start some chemical digestion. So we do have the pepsin enzyme in the stomach that breaks down proteins, so breaking the peptide bonds by pepsin. Peptide bonds? Pepsin? Okay, so there's your chemical digestion. So breaking down our protein. We also have some breaking down of fat in the stomach, but really we're just more so just breaking it down into smaller pieces. When our food is in the stomach, it's going to get broken down and eventually become chyme. So it's kind of the semi-digested liquid form of our food now that our stomach has gotten a hold on it. So even though we typically think of our stomach as our digestion center, it's actually, most digestion actually occurs in the small intestine. So our stomach's job is just to liquefy everything. Okay, so only thing I want to mention on this one is, again, you probably think we've already covered this. Yeah, because it's in the circulatory system. So blood supply to the stomach, we have our celiac trunk. Of course, you also get some blood supply from the splenic artery. And then once you leave the stomach, so we do have some absorption of nutrients in the stomach. That blood leaves the stomach and enters our hepatic portal system. And it's going to go into the liver, where the job of the liver is to have major nutrient storage. So even with the small intestines, large intestines, all of that stuff goes back to the liver. Your liver is able to detox stuff. You're able to store, for example, amino acids, glucose, helping to iron, helping to just act as a storage for what your body may need later on. So, inside the stomach, in our endothelial layer, we have what's called gastric pits. So, these pits are what's going to do the work of creating our different gastric secretions. So, we have different cells of our gastric pits. We have our mucosal cells. So, these mucus cells are going to create a mucus lining that helps to protect the stomach, since you are in a very acidic environment, so you don't want those cells to get damaged. Next type are regenerative stem cells, basically replacing cells that do die off, because again, your digestive system, it is exposed to the outside environment, so very likely to have damage, and so needs those cells to be replaced. Next cell, we have our parietal cells. These are very important cells because their job is to make the hydrochloric acid. So we'll talk about those steps later. But that's going to help to really break down the food with that strong acid. It also releases the hunger hormone called ghrelin, so helping to stimulate the brain. So going to go to the bloodstream and therefore going to the brain and telling the brain, hey, stomach is empty, so please feed it. Thank you. Okay, next one, we have chief cells. Their job is to make gastric lipase, so one of the enzymes to break down lipids. So ACE for enzyme, lip for lipid, and gastric because it's the one in the stomach. So enzyme to help break down our lipids. And also pepsinogen, which is the precursor to pepsin. So it's going to be just converted into the active form to break down our peptide bonds between our amino acids. And the last one are enteroendocrine cells. So these are hormonal cells that are going to help with regulating the digestion by sending both hormones into the bloodstream and paracrine signals to just nearby other parts of the digestive system. Okay, let's make some hydrochloric acid. So the combination of all of the different components of the secretions made in our stomach is called gastric juice. So it's part water, part hydrochloric acid, part pepsin, different enzymes in our stomach to help break down our food. So our parietal cells are going to be the one that are going to make our hydrochloric acid. And guess what? We're going to use that carbonic acid reaction again. Yay! Okay, so here's the reaction. Again, this is the chemical symbol one. So we have our carbon dioxide and water. We have our carbonic anhydrase enzyme, which also is in the parietal cells to help kind of speed the reaction along. That gets converted into carbonic acid, and then carbonic acid dissociates or breaks apart into its bicarbonate and hydrogen ion. So the bicarbonate ion, just like with your respiratory system, you have your chloride shift, so you have that exchange. So bicarbonate is pushed out into your bloodstream, your chloride is pushed out into your open lumen of your stomach, and it bonds with the hydrogen ion. Since chloride is negatively charged, hydrogen is positively charged, so opposite charges are attracted to each other, and so they're going to bind and form our hydrochloric acid. So this is just all the pieces yet again. So we go through our whole equation. The hydrogen ion is pumped into the gastric gland lumen, so open space of the gastric gland. Our bicarbonate is exchanged for chloride using chloride shift. So our positively charged hydrogen, negatively charged chloride ion are attracted to each other, and so therefore they're going to end up forming a bond and creating hydrochloric acid. So with our bicarbonate ion that was exchanged for chloride, because you now have this big flush of bicarbonate ions that have left the stomach and gone to our bloodstream, that creates what's called alkaline tide. So it's a high pH, so very basic or alkaline pH of the blood as it leaves the stomach. And eventually this is going to get diluted out, but there's still this initial wave of this basic or alkaline blood because you have all that bicarbonate. So thinking like you added a bunch of baking soda to the blood. Okay, so picture versions for those that like pictures. So our parietal cells are going to take carbon dioxide from the bloodstream. We go through our carbonic acid reaction. Then our carbonic acid ionizes into bicarbonate and hydrogen. The hydrogen is pumped out into our gastric gland lumen. Our bicarbonate goes through chloride shift, so we have an exchange with chloride. So we now have bicarbonate in the bloodstream. Chloride is removed from the bloodstream, pumped out into our gastric gland lumen. Negatively charged chloride, positively charged hydrogen. They're going to be attracted to each other because of the opposite charge. And eventually they combine and form our hydrochloric acid. Okay, so what does this hydrochloric acid do? So, not only is it going to physically break down food, but it also helps to activate our different enzymes. So, remember earlier our pepsinogen, it has to get converted to pepsin. So, that's the job of our hydrochloric acid. It's going to convert that precursor enzyme into our active form. Same thing with lingual lipase. So, lingual lipase does some work. It's okay in the mouth, but it's activated and even more efficient in the stomach because it's been activated by our hydrochloric acid. So, hydrochloric acid, very strong acid to help to break down our connective tissues and plant cell walls. So, something that is more difficult to break down. So, like, you know, they talk about different whole foods and whatnot. So, it takes a little bit longer on the digestion. So, you feel fuller longer, blah, blah, blah. So, that hydrochloric acid is going to help to break down stuff that just can't easily dissolve in water. Another function is to convert our ingested ferric ions to ferrous ions. So the iron that we take in to our body, that has to be converted in order to absorb that iron. So our ferrous ions so the ferrous iron, so ferrous as in referring to iron. So our iron ions, once it gets converted by our hydrochloric acid, it's absorbed, and then that iron can be used for hemoglobin, also for myoglobin, as well as our cytochromes. And it also helps with our immune system function since our acid is very strong. And so any kind of living thing is also going to be liquefied, just like our regular cells that we have eaten. So bacteria helps to break down, as well as breaking down viruses. So helping to stop them from causing a disease or some kind of infection happening in your body. So with our enzymes, some of them aren't in their active form once they've been first secreted out, and they're called zymogens. And so they're inactive. So they can actually be converted into their active form by making some adjustments to their actual structure. So, for example, removing some of their amino acids. So, for example, we have pepsinogen gets activated by our hydrochloric acid to turn into pepsin. And with our pepsin being activated, it also can help to activate other pepsinogen molecules. So that's the autocatalytic effect, where the activated pepsin can activate other pepsinogens into pepsin. So pepsin is going to do its job by breaking down protein. So if we're removing some of the amino acids off of pepsin, removing some of the amino acids off of pepsinogen because pepsin is doing its job, that also helps to activate more pepsinogen into pepsin. So pepsin's job is to break down our dietary proteins into shorter peptides. So we're not yet at the level where we're breaking down stuff into the monomer amino acids that we can absorb into the body, but we are on the way there. So we're just making our larger molecules into smaller molecules. Gastric lipase and lingual lipase, so breaking down lipids, those are just, again, we're breaking down our larger molecules into smaller, more easily to manage ones. So once we get into our small intestines, those enzymes can then break down stuff into an absorbable form. Okay, so let's move stuff around in our stomach, as well as making sure we can protect the stomach. So in our stomach, we do have a muscle layer with actually two different directions of muscle layer, and that allows for all of the churning inside the stomach to mix our food with our gastric juices. So these contractions, called peristaltic contractions, are controlled by our enteric pacemaker cells. So enteric as in referring to our enteric nervous system. So these just have our consistent contractions just to mix everything up consistently as we are breaking down our solid food into its liquid form. So helping with mechanical digestion. For the stomach, even though you are going to have some monomer building blocks being broken off just in that process, the stomach really doesn't really absorb too much. It can absorb aspirin. Some lipid-soluble drugs can also be absorbed in the stomach, but most of your absorption and breaking down or digestion of your food actually happens in your small intestines. So with our stomach, again, it is in an acidic environment. So how do we protect that sucker? So first we have our mucus coat. So the cells in the gastric pit, they make mucus to help to just provide a coating protection on those cells of the stomach to keep them from being broken down. There are tight junctions between our endothelial cells that help to prevent our gastric juice, or that acid, from exiting out of the stomach and into the rest of the body. And then eventually, of course, with our epithelial tissue, they're going to die off. And so we have those basal stem cells to replace the epithelial cells that die off. Okay, so let's look at regulating our gastric function. So again, part of it is our enteric nervous system, but then there's also communication between our digestive system and the brain, and different hormones that can be released that also control the function. So our gastric activity has three phases. We have our cephalic phase, which is controlled by the brain. So thinking of cephalic as referring to the head. Gastric phase, so that's your stomach, so it controls itself. And then we have our intestinal phase, so that's the intestines. So good job with that. So with our phases, again, as you eat you're consistently breaking stuff down and sending stuff to the intestines. And so this actually can happen all at the same time, because you're not just, oh, it's dinner time, here's my dinner pill, and you take that one thing and then you're done. You're eating over time. It's a process for your actual your meal time, if you will. Okay, so first phase, our cephalic phase, that's all about your sensory of food. So the smell, taste, or thought of the food. So basically, it's all in your head. So anything that comes into your brain that helps you to remind yourself of food, or if you're on Instagram or some other social media and you see someone's post of their meal, or a business advertising whatever, food that they might make, that sensory information comes in. Same thing like if you're at like an airport or at the mall, even though I know most people don't go to malls anymore, but oh well. So what, for example, like Cinnabon, what they'll do is they'll take their air outflow vent and it's right next to the oven. So the smell of freshly baked cinnamon rolls actually gets pumped out into the airport or mall space. And that helps to stimulate your digestive system, because it's helping to put input for our cephalic phase. And then even the thought of food, sometimes where someone might mention, oh, it's Tuesday, and you're like, oh, Taco Tuesday. And then it's like, oh, tacos would be nice. And that also helps to kind of get everything started for your digestive system, as in, yes, tacos would be nice. Okay, so sensory and mental input. So again, all the stuff that's happening in your brain. I don't care about you knowing about all the junk. Either way, I just want you to skip all that. So sensory and mental inputs that stimulates the enteric nervous system, therefore stimulating gastric secretion. So starting our process of our gastric juices being released in the stomach. So just in case you just happen to act on that sensory input of food, your stomach is already starting to be ready. So kind of warming everything up. Next phase is gastric phase. So, this is your stomach controlling stuff. So you now have actually eaten something to put into your food, into your stomach. So any kind of swallow food or semi-digested protein are going to activate this phase. So because you are taking input into your stomach, you're going to stretch the stomach a little bit. And so you're going to have this reflex of, oh, there's food in the stomach. So that's going to increase your gastric secretions being released, because now you actually need to have the hydrochloric acid and need to have your other enzymes released so you can break down the food that's actually physically in your stomach. So the pH is decreased, so that's because of the hydrochloric acid, and helping to break down the food that is there into the liquid form, into that chyme. The last phase is our intestinal phase. So hormones get secreted to help with this phase, as well as our nervous system, helping to control muscle movement. So your stomach's going to start contracting and moving food through into your small intestine. So it's little bitty tiny bits. It's not like everything just stays in your stomach and it just waits till, oh, it's finally, everything's liquid now. Now we can start releasing into the small intestines. Actually, as your stomach is contracting, it's pushing little bits of food that just happen to be liquefied already and moving that into your small intestines. So that's why, if you're, taking your time while you're eating or whatever, or it's Thanksgiving and, you start off with your regular lunch or dinner meal and you're still thinking about, ooh, that potato pie is still sweet potato pie or pumpkin pie, sorry, that's what I was thinking. It's still sitting on the counter and you're like, I kind of want that, but my plate, I got too big. And yeah, that's why I can, just take a little time for that digestion. And even though there's food still in your stomach, you can go ahead and get your pie. Okay. So also in this phase, we have the release of secretin, which is going to help with our pH regulation. Remembering our stomach is very acidic, and so as you're going into your intestines, that acid has to be neutralized. Cholecystokinin, it's another hormone that is released, so that's going to stimulate the release of bile from our gallbladder, as well as our pancrease secretion. So the stuff from the pancreas, such as our bicarbonate, that's going to help with regulating our acidic chyme, so neutralizing the acid in it, as well as our other hormones released from the pancreas, because now it's like, oh, we have actual food, so we need to tell the cells to actually do the job of absorbing the nutrients from the bloodstream. So another hormone that gets released is our, long word, glucose-dependent insulinotropic peptide, GIP for short, and that's going to stimulate the release of insulin from the pancreas. So glucose-dependent as in when you actually detect glucose in whatever you're eating, that's going to release this peptide. That peptide can then be used as a signal to tell the pancreas, hey, release some insulin because you need to tell the cells to absorb the glucose that's there. Now, granted, yes, the insulin's also going to stimulate the uptake of amino acids from proteins, but the main function is for glucose and regulating your blood sugar levels. Okay, we're going to start this one on a new video because it's a whole different section, so I will see you in the next video. Bye!